Hi, dear students. Welcome to one more session of genetics, and today we're going to talk about recombinant DNA technology or genetic engineering. If you look onto the term recombinant DNA technology, it's quite understanding that we are trying to make a recombinant. That is, we are taking different things and we're making a combination. And of course, when you say recombinant DNA technology, it implies that you're taking DNA and you're trying to prepare a recombinant. And since you are using DNA and you are doing engineering work, it is also called as genetic engineering. The basis of recombinant DNA technology or genetic engineering, it becomes possible mainly because the DNA molecule, let it be in a bacteria or let it be in a huge whale, the constitution of DNA or the basic composition of DNA is just the same. That is, they are all made up of nucleotide bases. They are all made up of 3-5- phosphodiester bonds. And all that is the same. Let it be a bacteria or let it be a whale. So, since the nucleic acids or the DNA, which has been found in all these organisms, are the basically the same. Of course, the constituents, the amount of nucleotide bases as well as the sequence, everything could be different. But in spite of this, they do share something in common, and hence more interest was being developed, like why not we engineer them. So let's see what is recombinant DNA technology or what is genetic engineering. If you go to look into a yeast cell, okay, a yeast cell is normally used to convert sugar into carbon dioxide and alcohol, and this is quite frequently used in baking as well as brewing. Now, what if you take this yeast cell and if you put some DNA into it and make it produce some vaccine for the human disease? Such a situation where one uh, living cell is being used to produce something other than its normal products. It could be products which are being of good interest for human value. Of course, we do things for our own value. So, if you make a living cell to produce other more valuable products, that is referred to as a in case of genetic engineering. Now, here in this example, which I am mentioning over here, you have an yeast cell, and this yeast cell can be engineered to produce a vaccines for human disease. That is a case of genetic engineering. So, genetic engineering is about changing the DNA of a living thing to change its characteristics. As we know, everything is being produced in a cell when it is being coded by a DNA. So, if you want to make something produce an unknown product or a foreign product from it, you have to produce the gene which is or the DNA which is responsible for the production of that particular product. So in this case, the vaccine, if you're considering if you're thinking about COVID-19 itself, some of the proteins or some of the immunogenetic elements of COVID-19 could be, or the genes which have been coding for the immunogenic elements of COVID-19, that could be what they could be injected into the yeast and we could make it produce that particular element. I just took COVID-19 as an example because it is a contemporary example. There are different things. We already have various vaccines in which various proteins as well as various elements which are being considered to be as immunogenic are being produced by genetic engineering. Now, what is a recombinant DNA? A recombinant DNA, or shortly we also call it as a rDNA, It is a term used to describe the combination of two strands that are constructed artificially. Now, if you take a bacterial DNA and if you take a DNA from human, okay, it's not the whole DNA, you take a small portion of that, and if you chop that and if you make them combine together and form a artificial DNA, that is a recombinant DNA. Like naturally occurring DNA, the recombinant DNA also has the ability to produce the recombinant proteins. And it is often these proteins that play a key role in the application of recombinant DNA. Now, let me just give you some examples of recombinant DNA. As I told you here, if you take the bacterial DNA, that is, I'm taking a bacterial some DNA, and you take the human gene for insulin. So, my interest of the protein of interest, which I need to produce here, is insulin. You know what is insulin? Right. Okay, fine. So the human gene for insulin is taken and the bacterial DNA is also being taken and that has been combined together. So I will get an artificial DNA which will code for the human gene of insulin. Or you can also take a plant DNA and a bacterial DNA. Now, for example, from Agrobacterium tumefaciens is a bacteria which grows, which can infect normally itself in the case of plants. So when two different types of DNA are being combined together, you form the recombinant DNA. Or you can also take a mouse DNA and combine it with a human DNA. These two things together will form a recombinant DNA. So, in recombinant DNA technology or genetic engineering, it could be any two different DNA which have been combined together so that and this DNA is being combined together, and once the recombinant DNA has been formed, this recombinant DNA will be given to a particular uh, what is it, you will put it into a particular host so that this recombinant DNA will be expressed over there. Mm. So the purpose of producing recombinant DNA is so that we want to combine what, our gene of interest with something else. Now, what is the purpose of doing such a thing? Okay, if I give you an case, you can see you need to produce human insulin in large amounts. Of course, humans, we already produce human insulin. But the amount of insulin produced in certain people are being found to be very less. And that is a reason probably that could lead into diabetes and all that. So, what we try to do is you take the particular gene which has been coding for human insulin and combine it you combine it with the bacterial DNA, and then what happens, bacteria will multiply at a very rapid rate and you make the bacteria to produce the human insulin. Okay, and then we extract the insulin and we use it as a pharmaceutical agent. So the whole purpose of recombinant DNA technology is to mainly produce a large amount of our products of interest. Okay, it could be of different things. Now, insulin is just in one example of that. We will be coming across to the various examples of that in the coming classes. But the whole purpose of recombinant DNA technology is to produce our product of interest in large quantities when the normal product is produced only at a small rate in a normal situation. Okay, so the product of recombinant DNA technology is something which has been produced of very much importance and which need to be produced at a very faster rate. So the gene which is coding for that particular product is being combined with another gene, okay, another gene, in a sense, a carrying agent. Now it could be a bacterial DNA, it could be a viral DNA, or whatever it is. You combine it and you make it as a recombinant DNA. Now in this picture, you consider, now the gene of interest here. This is a cell containing the gene of interest. You take the DNA from it and you cut the DNA. See, look at the red color over here. This is the gene of interest. I take the gene of interest and I told you, you have to combine it with something so that a recombinant DNA has been formed. So, okay, here I'm taking a bacterial plasmid. Now, what is a plasmid? You should know something. This gene of interest can be combined only with something which is already capable of or which could be cut, and at the same time which is ready to carry small amount of DNA. So now this is called the vector. The gene of interest is combined with the vector. The details of the vectors and all that I will be telling here. I'm just giving you a brief introduction right now. So I took my gene of interest and I combined it with a vector DNA, or the one which will code my gene or which will carry my gene of interest. Now here the vector here is a plasmid, and the vector is in such a way that it was able to multiply rapidly, and it is having certain other properties also. Let's go into the details later. So you take the vector DNA, that is a plasmid over here, and I cut the vector as well as I cut the gene of interest, and with some enzymes. They are called as restriction enzymes, and you put the vector as well as the gene of interest so that they combine together to form a recombinant DNA over here. And once the recombinant DNA is been formed, this recombinant DNA is put into a host cell. What is a host cell? The host cell is a one which will carry the recombinant DNA. So the recombined DNA, that is a plasmid and the gene of interest together combined, they are put into a host cell. And here, since the plasmid can replicate in a bacteria, we have taken a bacteria as a plasmid. It can be anything. If the recombinant DNA can multiply in a eukaryotic cell, you can put it into a eukaryotic cell also. But since in this example we have taken a plasmid, this recombinant is put into a bacterial plasmid. Into a bacteria, sorry. And once the recombinant is put into the host cell, bacteria here, this is the example of a bacterial host, the bacterial host will multiply and as a result, what happens, the gene of interest also will be produced in large amounts, and it will produce different copies of the particular gene, and the products of that particular gene would also be produced. And these proteins, okay, or the products of that particular gene could be of different types. Either it could be a gene for a pest resistance which is inserted into the plant, or it could be a gene which is used to alter bacteria for cleaning up toxic waste, or it could be any protein which has been used to dissolve the blood clots in a heart attack therapy, or it could be any protein which has been used to make snow form at higher temperature. So the gene of interest could be of different reasons. Okay, it could be of therapeutic importance, or it could be of a bioremediatory response, or it could be something which would help us to promote agriculture. Whatever be the reason, the basic concept of recombinant DNA technology is the gene of interest is being cut from its correct location, and it has been combined with a vector molecule. Vector molecule is something which can carry your gene of interest, okay, and they are glued together using certain enzymes called the ligases, and then as a result you will get a recombinant. This is a recombinant DNA, and once the recombinant DNA is there, the recombinant DNA is inserted into a host cell, or host cell is something in which the recombinant DNA can multiply as well as express. Now, since we have taken plasmid DNA in this example, this recombinant DNA is put into a bacterial cell here, and that is what we have showing, the recombinant bacteria. And when this recombinant bacteria multiplies, what happens? The gene of interest also multiplies, and the proteins which are being coded by the gene of interest would also be expressed, and that would be further extracted and purified for the use of different reasons. I hope you got a brief outline of what is recombinant DNA technology, the examples, as well as the what are the main steps of it. We will be continuing in the next sessions. Thank you for now.